Welcome back, ladies and gentlemen. So today we're going to talk about the Calvin cycle, or you may come across it as the dark reactions or the light independent reactions. All of those terms can be used interchangeably. I've got Calvin cycle here, light independent reactions here, dark reactions here. All of those things mean the same thing that we're going to talk about today. We tend to try to stay towards that Calvin cycle now, mostly because using the terms dark reactions or like independent reactions kind of gave it some misinformation or miscommunication where people were thinking that these have to happen at night while the light reactions are all happening during the day, and that's not really true. Um, all it means is that the light reactions require light to be present in order to function, whereas the Calvin cycle will be able to continue to function, at least for short stints of time, without light present. That being said, these don't necessarily just happen at night. They are happening during the day when the light is out, when the sun is out, and can only function for short periods of time in the dark because eventually we're going to run out of that ATP and NADPH that are coming as a result of those light reactions. So we're going to try to stick to this term, but I throw those other terms out there just in case that you see them. So the Calvin cycle is going to occur in the stroma, and we're going to use the ATP and NADPH from the light reactions to drive this cycle, as well as needing the input of carbon dioxide. And ultimately, we're going to produce sugars, specifically in this sugar precursor molecule, something called G3P, which we can then put together multiple G3P molecules to make glucose, as well as other molecules that the plants might need that have a sugar derivative. So the cycle is going to consist of three phases. Unlike the Krebs cycle in glycolysis, I am going to ask you to know a little bit more about the Calvin cycle in terms of what these phases are and the basics of what happens. So we're going to take a couple of minutes here and go through what this is. So first phase is going to be carbon fixation. We're going to fix or steal that carbon off that carbon dioxide molecule and keep it and use it. We're then going to go through a reduction phase, which will be phase 2. Part of that reduction phase will release some of this molecule called G3P, or glyceraldehyde 3-phosphate, if you want to know what the full term is. But nobody wants to have to write out glyceraldehyde 3-phosphate, so they shorten it to G3P. This is that sugar precursor that we can then build into things like glucose. And then phase 3 is going to be the regeneration of this molecule called RUBP. And that RUBP molecule is going to be really important because it's a carbon dioxide acceptor. It's going to be what allows us to steal that carbon dioxide out of the plant's environment and actually be able to harvest the carbon from it. So let's look at how this works. Okay, so a couple of things that you should know in here in terms of this diagram. It's a little kind of crazy, a little complex, but let's break it down. So this first part here is your carbon fixation. And so here's this RUBP molecule right here. And if I look, each of these dots is a carbon. So if I count 1, 2, 3, 4, this is a 5-carbon molecule. Okay, I'm going to bring in my carbon dioxide from the environment, and here is that G3P molecule here. Okay, it's a 3-carbon molecule. Okay, that will ultimately create that G3P down here, which is also a 3-carbon molecule. Okay, so we're going to trace carbons here. So I'm going to take this RUBP, which is a 5-carbon molecule. I'm going to bring in carbon dioxide, and I'm going to allow that single carbon to get attached to that RUBP, so it makes a really short-lived 6-carbon molecule here with the help of this enzyme called ribisco. Okay, so this guy's an enzyme. Okay, so 5-carbon, take that carbon off the CO2. I now have a 6-carbon molecule with the help of that ribisco. It doesn't last very long, and so we're going to quickly break it into 2-carbon molecules here. That's going to bring us over into this reduction phase. So here I have 2 of these molecules here, right, 3 carbons each, okay, and with the help of ATP and that NADPH that we had from the light reactions, that's going to ultimately create that G3P molecule. Okay, and that's the point of this reduction phase. So taking that 3-carbon molecule, rearranging, utilizing that ATP and NADPH to ultimately put it into this form of G3P. Now, some of that G3P is able to come off of the cycle, but if you look, most of the G3P that we create ends up in phase 3. And phase 3 takes a lot of that G3P as well as some of that ATP. Okay, so G3P with the help of the ATP to then rebuild that 5-carbon sugar molecule, that RUBP, so that the cycle can keep going. Now, if you take a look at this, G3P is a 3-carbon molecule. For every carbon dioxide that comes in, I'm only bringing in a single carbon. So what that tells us is that in order to make this cycle work, I need 6 carbon dioxide molecules for 1 glucose. So 6 turns of the cycle, meaning 6 CO2 molecules in order to make a single glucose, right? Remember, glucose is C6H12O6. So I need those 6 carbon molecules here, those 6 carbon atoms, in order to make that 6 carbon in a single glucose. That also means that in order to make this work, I actually need 2 G3Ps to make a single glucose molecule, because remember those G3Ps are 3 carbons apiece. So in order to make this work, there's where those 6 carbons in that overall chemical reaction for photosynthesis, I need 6 of them in because I need 6 carbons to go through this cycle to ultimately make me 2 G3P molecules, which then can get put together to make a single glucose. So the G3P then that isn't being given off again goes back through with the help of some of that ATP to rebuild that RUBP molecule so that I can then continue to use that and the enzyme Rubisco to continue pulling off carbons from carbon dioxide and keep the cycle going.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of 2 sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem II that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.